Hi everyone, welcome back to my channel. In this video, we shall learn about two concepts. One is active site of the enzyme, and the second one is the most important one with respect to enzymes, that is enzyme specificity. And we said that enzymes will act as a biocatalyst. They alter the rate of biochemical reaction by carrying out the reaction in an alternate path by lowering the energy of the reaction. That's the thing we already said in our previous video. But if you compare the substrate molecule and the enzyme molecule, the size of enzyme molecule will be much more bigger compared to that of substrate molecule. So we will be having much of surface area for the enzyme molecule. And in order to convert a substrate molecule into its corresponding product, the substrate molecule is supposed to accumulate, adsorb or settle on the surface of the enzyme. But all the regions present on the enzyme surface, I repeat, all the regions present on the surface of the enzyme will not be active or will not be efficient so as to receive the substrate molecule and thereafter converting the substrate into product. That means all the positions on the surface of the enzyme molecule are not effective or not active to receive the substrate molecule so as to carry out the reaction. Only certain positions, certain specific positions of the enzymatic surface are capable of receiving the substrate molecule and then thereafter converting that substrate molecule into product. And that region on the enzyme at which binding of the substrate molecule can occur and thereby catalyzing the conversion of the substrate into product will be possible. And that region at which the substrate molecule actually settles, accommodates, and that region is called as active site of the enzyme. Okay, so active site is the region on a enzymatic surface where the binding of substrate molecule can happen so as to convert into product. That is the simplest definition for active site. And we already said that the enzyme is having two parts. One is the proteinish part, that what we call as apoenzyme, and non-protein part, that what we also call as coenzyme. So in enzyme we are having proteins, that is apoenzyme, right? So in enzyme, since it is a protein molecule, we'll be having more number of amino acids which are joined together through peptide bonds, that we are well aware how the protein molecule will be formed by the combination of a peptide bond. So on the surface of the enzyme molecule, we will be having peptide bonds, as well as peptide bonds nothing but we will be having amino acid residues. So at the active site, the amino acid residues will specifically bind the substrate molecule by means of non-covalent interactions, not through covalent interactions, it is through non-covalent interactions. I repeat, at the position of active site, the amino acid residues which are present will combine or will bind the substrate molecule. The amino acid residues present at the active site will bind the substrate molecule through non-covalent interactions, and because of this, that results in the formation of an enzyme substrate complex. Enzyme substrate complex. And the formed enzyme substrate complex will make the substrate molecule so as to react with each other. That means the enzyme molecule, by receiving the substrate molecule onto its active site, makes the substrate molecules to break the older bonds and to form the newer bonds between the substrate molecule in the enzyme substrate complex, and the substrate molecules will self interact with each other so as to convert into product. So once the product is formed, the product will comes out of the enzyme surface, so that the enzyme will get regenerate and it get its original structure, and once again the enzyme will be ready for receiving the substrate molecules. So in this way, the active site will activate the substrate molecule to form an enzyme substrate complex, which upon decomposition converts into product. But all the amino acid residues that are present in the protein part of the enzyme will not perform this function. Only the specific or unique amino acid residues. That means at the active site, the enzyme represents a unique three-dimensional region that involving the amino acid residues, and those amino acids that are present in the active site can only bind the substrate molecule through the non-covalent interaction. And you may get a doubt, so since we are having much of protein part in the enzyme, so if only the amino acid molecules or amino acid residues that are present in the active site are binding the substrate molecule, what about the remaining portion of the protein part, that of the apoenzyme? By leaving the amino acid residues that are present at the active site, the remaining polypeptide chain of the protein part of the apoenzyme will helps in maintaining the required three-dimensional conformation for the active site. So in this way, the whole and soul components of the enzyme will perform their own works so as to make the enzyme more active, so that it can receive the substrate molecule, absorb the substrate molecule, and that can convert into enzyme substrate complex, as I said, which upon decomposition results in the formation of products. And now we shall see how all these things will actually happens at the active site. So we said the enzymes are the biocatalyst, which are compulsorily required for a biological reactions for the purpose of speeding up of the chemical reactions which can be conducted by the enzyme. And we said the polypeptide chain will give the three-dimensional conformation for the active site of the enzyme molecule, which receives the substrate molecule so as to fit in the active sites. So once the substrate molecule comes and fit in the active sites, that forms an enzyme substrate complex, and once the enzyme substrate complex is formed, the enzyme will make the substrate molecules to react with each other, thereby breaking and forming the bonds so as to convert the substrate molecules into product. Once the product formation is completed, the product will be coming out of the enzyme, and enzyme will take its original part and will be ready for receiving the substrate molecules again. We shall go on to the next topic, that is enzyme specificity. Like inorganic catalyst, the biological catalyst, that is enzymes, are also highly specific in nature. That means each and every enzyme is applicable or can catalyze only reactions are of only one type. Certain enzymes are applicable only for certain substrate molecules only. All the enzymes are not applicable for all the substrate molecules. Likewise, each and every enzyme molecule, each and every enzyme will be specific towards either particular functional group or particular bonds or particular type of reactions. Like this, each and every enzyme will show more specific nature towards catalyzing the biochemical reactions. And we can say the specificity is one of the major criterion of the enzyme action. But if you come to the case of enzymatic action, that is specificity, there are several various types of specificities will be exhibiting by the enzyme molecule. And now we shall see what are those different types of specificities that an enzyme molecule do possess. So in that, the first type of enzyme specificity is called as reaction specificity. Reaction specificity. So the name itself telling you that mean when you are having the same substrate molecule, but when you are using different enzymes as catalyst, then though we are using the same substrate molecule, the product formation will get differ depending upon the nature or type of the enzyme we are using for that substrate molecule in order to convert that into reaction. So let us consider one simple example. Let us consider an amino acid molecule COOH NH2. So we have considered an amino acid molecule, and when on this amino acid molecule, if you perform the catalysis by the enzyme oxidase, the name itself telling you this is the enzyme which perform the oxidation. So when this amino acid molecule is oxidized in presence of the enzyme oxidase, that results in the formation of RCOCOOH with the elimination of NH3. So ammonia got eliminated. Suppose on the same amino acid like RCH COOH NH2, I have considered the same substrate molecule, but instead of oxidase, if I have used another enzyme decarboxylase, if I use the enzyme decarboxylase, then the name itself telling you it performs the decarboxylation, so that we will be getting the product RCH2 NH2 with the expulsion of carbon dioxide. So that means reaction specificity in the sense different enzymes, when they're acting on the same substrate molecule, they bring out different reactions and thereby produce different products. That's about reaction specificity, so that we can say. Next, number 2, it is most important one, that is substrate specificity. Substrate specificity, as the name itself telling you, this specificity of enzymes is towards the substrate molecule we are using in the reaction system. And this substrate specificity is having several types. In that, first one is absolute specificity. Absolute specificity is the first type under substrate specificity, and the name itself telling you absolute specificity. That means in this absolute specificity, some enzymes will catalyze only one type of reactions. Only one type of reaction, or if it is specific towards only a one particular type of bond, that is RA, or towards a particular bond type. Particular bond type, then such kind of specificity exhibiting by the enzymes will be called as absolute specificity. And here we can say absolute specificity means they will catalyze only one type of reactions or towards a particular type of bond, or they show specificity towards particular substrate molecule. They show specificity towards a particular substrate molecule. That means one enzyme can act on only one substrate molecule, not on all the substrate molecule. One enzyme can act on a particular type of bond, not on all the various types of bonds. And one enzyme can catalyze the reactions of only one type and not on all other types of reactions. And now we shall consider some simple examples so as to give justification for this particular thing what we said under absolute specificity. So if you consider the enzyme urease. Urease is the enzyme which can perform the hydrolysis of urea only, and this enzyme urease won't take part in the any biochemical reaction other than hydrolyzing the urea. And that's why we said the enzymes will be specific towards a particular substrate material, particular substrate molecule, not on all substrate molecule. That's what we said. Urease is the enzyme which can catalyze the hydrolysis of only urea. So we know that when urea is hydrolyzed in presence of urease, we will be getting mixture of carbon dioxide and ammonia. We cannot use this enzyme urease for performing catalysis of any other reactant molecule, or in simply speaking substrate molecule. So one more example we can give, like SDH, called as succinate dehydrogenase. SDH is called succinate dehydrogenase. So this particular enzyme can perform on only one particular material, that is succinate, and not on urea. SDH cannot be used on urea, and urease cannot be used on succinate. So that each and every enzyme is specific towards a particular substrate molecule. So when SDH is acted on succinate in presence of a receptor such as FAD, called as flavin adenosine or adenine dinucleotide. Flavin adenosine dinucleotide is the full form of FAD. So succinate in presence of the receptor FAD and catalyzing by the catalyst SDH, that is succinate dehydrogenase, reversibly the reaction will produce fumarate. And since we are telling it performs the dehydrogenation, that will be received by the FAD molecule, that we will be get FADH2. This is the reduced form of the receptor FAD, that is flavin adenosine dinucleotide. So here we observed the specificity of the enzyme towards the substrate molecule. So in case of absolute specificity, enzymes not only show specificity towards the substrate molecule, they are specific towards the particular type of bonds also, and that also we shall see under absolute specificity, that is bond specificity. So here if you consider certain enzymes, they are specific towards the substrate having similar bonds and similar structures. That means each and certain enzymes are specific with substrates having, they are specific towards the substrate molecules having similar bonds. And since this bond specificity is common towards the substrate molecules having a similar type of bonds, this bond specificity is also called as relative absolute specificity. Relative absolute specificity is the other name we can give for bond specificity. So for example, the enzyme amylase. This is the enzyme which can hydrolyze alpha 1, 4 glycosidic linkage or glycosidic bond alpha 1, 4 glycosidic bond in both starch and glycogen. So both in starch and glycogen, amylose can be used to perform the hydrolysis of alpha 1, 4 glycosidic bond. So in both of these materials, in order to break alpha 1, 4 glycosidic bond, we can use amylose itself. And one more example we can give, that is lipase. Lipase is the enzyme which will be used to perform hydrolysis of ester bonds. Lipase is the enzyme which is used for the process of hydrolysis of ester bond, which is formed between glycerol and fatty acids. Lipase is the enzyme which is used for the hydrolysis of ester bond between glycerol and fatty acid, which is present in any fat material. So here also we are looking the common thing. So wherever the ester bond is there, which is formed between glycerol and any fatty acid, lipase can be used, that ester bond, lipase can be used to break that ester bond which is present in a fat material. And that's why we can say the relative absolute specificity, that are also called as bond specificity in case of amylose and lipase. Next, the second absolute specificity under the second substrate specificity is called as group specificity. Group specificity. So the name itself telling you, this is the specificity showing by the enzyme molecules on a group. That means now the second specificity under substrate specificity is group specificity. The name itself telling you, this is the specificity showing by the enzyme molecule on a group. That means when the enzyme molecule act only on molecules that have specific functional groups. When enzyme is acting or catalyzing on a particular type of functional groups, then such enzymes are said to be having group specificity. So for example, we are having an enzyme molecule X, and that will be having influence towards catalyzing towards only the alcohol molecules, alcohols, not on phenols, not on aldehydes, not on ketones. It will catalyze only alcohols. Likewise, each or certain enzyme molecules will be having influence or will be having catalytic activity towards a particular functional group. Such specificity is called as group specificity. And we said the bond specificity as relative specificity, and this group specificity is called moderate specificity. Group specificity is also called moderate specificity, and in sometimes it is also called structural specificity, because this also depends. That means the group specificity also depends how a functional group, the groups surrounded by a particular functional group. Based on that also the group specificity depends. That's why the group specificity is also called moderate specificity or structural specificity. And now we shall look over into some of the examples where we can observe the group specificity. If you consider an enzyme called hexokinase. If you consider an enzyme hexokinase, this is the enzyme which can be used for reaction with D-hexose. This hexokinase can be acted on any hexose sugar such as glucose, fructose, galactose. Like this, we can use this hexokinase on any D-hexose sugar when it combined with a high energy component such as adenosine triphosphate. Phosphorylation of hexose sugar will takes place, and that results in the formation of D-hexose 6-phosphate. So one phosphate have been transferred to hexose, so that ATP will get reduces to ADP, that is adenosine diphosphate. So hexokinase can be used for the phosphorylation of any hexose sugar, that what we are observing, the group specificity. Okay, and one more example we can give, that is ADH, called as alcohol dehydrogenase. The name itself telling you this is showing group specificity towards alcohols. So when on alcohol this ADH is acted, alcohol will be converted into corresponding product. So that we shall see how the reaction will take place. So when an alcohol is made to react with the ADH, that is alcohol dehydrogenase, in presence of a receptor such as NAD. We already said NAD is nicotinamide adenine dinucleotide. So alcohol in presence of a receptor NAD under the catalytic activity of alcohol dehydrogenase, alcohol will get, and in biological system, alcohol in the sense we will be encountering with only two alcohols, so that is methanol and ethanol. So when alcohol comes in contact with the ADH in presence of NAD, it results in the formation of an aldehyde, and NAD will get reduces to NADH with the expulsion of one proton. So here also you are observing the group specificity, that is this ADH can be used on alcohol, both methanol and ethanol, so as to get aldehyde. And likewise we can give few more examples for this group specificity, that is if you consider the two enzymes, so one is called pepsin. This pepsin is the enzyme, pepsin is the enzyme which can be used to perform the hydrolysis of the peptide bond, in which the amino group is contributed by the aromatic amino acid. So when amino acids are joined, we will be getting two ends, one is N terminal and C terminal, right? So if a peptide bond is formed by two amino acids, and if the amino group contribution is given by an aromatic amino acid such as phenylalanine, tyrosine and tryptophan. So when these three amino acids, that is phenylalanine, tryptophan and tyrosine, when these three amino acids were given contribution of amino group in developing a peptide bond, in breaking that peptide bond, pepsin can be used. So pepsin can commonly breaks the peptide bond whenever we are having tryptophan, tyrosine and phenylalanine. So here also pepsin is showing the group specificity. And one more example we can give, that is trypsin. Trypsin is the enzyme which can be used for hydrolysing the peptide linkages, in which the contribution of COOH group is given by two enzymes, that is arginine and lysine. When COOH contribution is given towards the formation of peptide bond by these two amino acids, then in order to break, or if we want to break that peptide bond, then we can use trypsin in both of these cases. So here also we are observing group specificity. Next, the last type of substrate specificity is stereospecificity. So the name can give certain or some information regarding the core meaning of this particular specificity. That is, certain enzymes will be specific towards a particular type of stereoisomer. That stereoisomer may be a geometrical isomer or a particular configuration, or even it may be a particular optical isomer, or even it may be a positional isomer also. Except the isomerase enzyme. Except the isomerase enzyme, majority of the enzymes will show this stereospecificity. That means this is the specificity showing by the enzyme molecules towards a specific type of stereoisomer. That may be a positional isomer, or maybe a geometrical isomer, or maybe a configurational isomer also. And we shall consider some simple examples in order to explain the concept of stereospecificity. If you consider this LDH, that is lactate dehydrogenase. Lactate dehydrogenase enzyme. This lactate dehydrogenase enzyme will act on this particular reactant molecule, that is substrate molecule, CO minus. So this is the lactate molecule. Here you can observe the hydrogen atom is present on the right hand side of the Fischer configuration. So this is having L configuration. So we can see it as L lactate. So this LDH, that is lactate dehydrogenase, will perform, the name itself telling you, dehydrogenase. So from this particular material, that is L lactate, it can remove the hydrogen. But remember, it coming under stereospecificity. That means this LDH can perform the dehydrogenation only on that lactate molecule which is having L configuration. LDH is inactive towards D-lactate. It won't act on the D-lactate. It is specific towards only L-lactate molecule. So when this L-lactate is reacted with NAD, which is the receptor, in presence of the enzyme LDH, that is lactate dehydrogenase, it results in the formation of this one with the production of reduced form of receptor, that is NADH and H+. And this is the pyruvate molecule. If you perform, you can observe the reaction is reversible in nature. When pyruvate molecule is reversibly trying to change the lactate, this pyruvate in presence of LDH will produce L-lactate only, but not the D-lactate. So here you can observe the specificity of this LDH towards a particular configuration, that is L configuration. In previous case, I said the stereospecificity towards particular configuration. And now we shall consider one more enzyme such as fumarase. So this fumarase enzyme can act on this thing, COO minus, that is fumarate. And here you can observe fumaric acid is having trans configuration, that is fumarate is having trans configuration. So when you perform the hydrolysis of this fumarate, the reaction can occur reversibly, and this results in the formation of CH2COO minus CHOH CO minus. Here also this is malate, but H is present on the right hand side of this chiral carbon atom in the Fischer projection. So this is having L configuration. But here you can observe both of the things. When fumarase is using as the enzyme to act on fumarate, so having trans configuration, upon hydrolysis it will produce only L maltate. But if this particular structure is having cis configuration, that is malate, malate on hydrolysis will not produce the malate, malate will not produce the L-malate. That means fumarase cannot act on the cis configuration of this particular structure. Means here also we observing the enzymes are specific towards a particular geometrical isomer. So this fumarase is particular towards the trans configuration of this structure, not on the cis configuration. So in this way, we can say that enzyme molecules are having specificity towards a particular type of bond, towards a particular type of functional groups, toward a particular type of isomeric form. So that is about specificity of the enzymes. Thank you.